Camera is here.
But it's anyway, it's good to see you.
Otherwise, it's just that people don't see. There's only the slide and
You're going to start because I'm going to add something.
Morning, everyone. So thanks a lot for attending this morning's seminar. So as usual, we are on webinar mode. So people online, don't hesitate to tell us any issues in the chat, and please ask your question in the Q&A tab. And we are aware that there's a slight issue with the camera now in the room, but we're just going to start. And if you give us a minute, it's going to be solved. And with that, thanks a lot for introducing to the speaker.
I want to introduce you Martin Kapun. He is evolutionary biologist, and he worked mostly in adaptation to temperatures, mostly in Drosophila, I think, but in other species too. He started his work as PhD student in Vienna. He made his PhD with Christian Schroeder. And after this, he moved to Lausanne, and he worked in adaptation of Drosophila to temperatures in inversion polymorphisms. And then he moved to Zurich for postdoc too. And he is co-founder of a big consortium it's called Droso-EU. And in frame this consortium, people want to understand the influence of different factors for Drosophila melanogaster, and Martin made a lot of work in this direction. Now he is working in Natural History Museum of Vienna, and he moved in his work to host pathogen host-symbiont interaction, and he will present his work today. And now he moved to museomics and to understand how we can use data from museums to understand adaptation. Yes, and now Martin will present his work.
Sveta, thank you very much, and thank you all for the invitation. It's really a great pleasure to be here with you, and it's always exciting to be in Montpellier, and I'm thrilled to be here. Yeah, thank you for the introduction, Sveta. You already presented like the big scope of my research interest, so I think we all agree that organisms living in heterogeneous environments are constantly challenged by different selection pressures that may eventually lead to adaptation, speciation, but also to extinction eventually. However, evolution is not necessarily driven by selection alone, but also by demographic effects. And as you all agree, it has proven pretty difficult sometimes to disentangle the relative effect of each of these mechanisms. And for me as an evolutionary biologist, it is my major goal and my major interest to better understand the evolutionary mechanisms that maintain and shape genetic and phenotypic variation in natural populations. And I'm using a combination of different approaches to tackle these questions. So let's see if this works. Yes. So needless to tell you, I guess, Drosophila melanogaster has proven very powerful, a powerful system to tackle these questions. And this is not only because it's a very well-established and long-standing genetic model, but also because it is characterized by a really very interesting evolutionary history. So as a human commensale, it originated as us humans from sub-Saharan Africa and only subsequently in multiple waves colonized and conquered the world. And since it's a fairly tropical organism, it had to adapt to very different climatic regions and acquired adaptations such as diapause propensity, which are very well understood and therefore make this very versatile model organisms to address fundamental questions in evolutionary biology. And today I want to tackle or to introduce 3 different topics that have been very important in my past research career. These topics range from population genetics to host-symbiont interaction, as I've already shown in my title. So Drosophila melanogaster occurs along environmental gradients, such as this temperature gradient along the North American East Coast. And these temperature gradients have long been in the focus of evolutionary biologists because they really represent natural test beds to study the evolutionary underpinnings of local adaptation. And particularly in Drosophila melanogaster, several gradually changing genotypic and phenotypic variation have been found along these environmental gradients, and these are called so-called clines. And these involve, for example, body size variation, development time variation, and so on, but also on the genetic level, allozyme variation, SNP frequency differences, and inversion polymorphisms. While these gradually changing phenotypes and genotypes are often considered to be prima facie evidence for locally spatially varying adaptation or selection, there are of course other evolutionary mechanisms that can result in fairly similar patterns. And these include, for example, isolation by distance, where just organisms have reduced dispersal abilities, which results in the building up of differentiation. Then range expansions, alleles may serve as a population expands simply because of chance, because an allele in the wavefront is very frequent, or due to secondary contact, when previously allopatric populations come together. Is everything that can be seen here? Okay, I think this works. Okay, sorry, I don't know why this doesn't work. Where am I? Okay. How can I? Here, I think now I am here. Right? Yes. So, okay. Some of the open questions related to clinal variation are, how is genetic variation structured along these environmental gradients? What are the underlying evolutionary forces? And what is the impact of clinal genetic variation on the adaptive phenotypes? One particularly well-known polymorphism that shows clinal patterns are chromosomal inversions. And these, as I don't need to tell Mathieu, are structural mutations that result in the reversal of gene order in the affected genomic region. And due to this change in the gene in the synteny, relative recombination rates are suppressed, and it is assumed that inversions can actively contribute to the adaptive process by keeping together co-adapted alleles and act as so-called supergenes. In a recent meta-analysis, we analyzed more than 500 population samples from all over the world, where we were able to identify. Where we were able to assess inversion frequencies of one particularly common inversion, which is called iron-3-apane. This is basically my pet inversion. And we identified that this inversion really occurred in clinal patterns all over the world. So you can see the inversion is fairly common in tropical environments and decreases in frequency, as highlighted here by this color gradient, towards higher latitudes in the northern hemisphere, but also reversed in the southern hemisphere. And this, of course, strongly points at an adaptive effect of this inversion. However, it remains unknown whether these adaptations are unique in the different environments, or whether this inversion has captured an adaptive phenotype and dragged it along as Drosophila spread across the world. And to address this question, we opted for a phylogenetic approach, and we combined more than 400 haplotypes from Drosophila melanogaster individuals collected all over the world. And this a neighbor joining network that you see here, you see that the leaves are color-coded according to the origin of these haplotypes. And you can also see, I hope this is visible, the edges of this network are color-coded according to the carrier type, whether they are of standard arrangement or whether they are carrying iron-3-apane. And in this first network, we focused only on SNPs located outside of the inversion, so not associated with the genomic region. And you can see a very star-like topology, which indicates a recent range expansion, population size expansion that predated the out of Africa expansion, and consistent with this African origin of Drosophila melanogaster, we can see that all non-African or most non-African flies form a monophyletic cluster in here. If we, however, focus on SNPs that are located inside the inversion, we can clearly see that they form a monophyletic cluster right within Africa, indicating that iron-3-apane is of African origin and carries along African genetic variation outside of Africa. And as you can see, there is nevertheless some population substructure in this monophyletic cluster, which may be the result of local adaptation to the different environments or due to founder effects. That's something that is not entirely clear. To better understand the impact of this inversion on genetic variation, we used pool sequencing data collected along the North American East Coast from 28 population samples and investigated to which extent genetic variation is associated with latitude. So in this so-called Manhattan plot up here, you can see that every SNP represents one data point, and on the y-axis, the negative logarithmic p-value is shown. And needless to say, one very giant region, that is the genomic region spanned by iron-3-apane, contributes most to genetic differentiation along the North American East Coast, indicating a very prominent role of the inversion along this gradient. We developed inversion-specific marker SNPs that are in very tight LD with the inversion breakpoints in order to identify the inversion frequencies, even if we don't have direct possibilities using, for example, cytological methods. And using these diagnostic marker SNPs, we were able to use these pool sequencing data to identify the inversion frequencies along the North American East Coast. And as you can see here by this line indicated in red, the inversion frequency is very consistent with previous data and shows a very steep climb from Florida that quickly decays towards northern latitudes, indicating that this inversion is very stable across more than 40 years of observation, which may further indicate that it has an adaptive effect. We were asked whether these patterns can be explained by neutral evolution alone, potentially due to isolation by distance. And we therefore sampled 20,000 SNPs located in short introns, which are assumed to evolve fairly neutrally, and investigated here, indicated by this black line, how the allele frequency change from north to south. And as you can see, the allele frequency change across these 1,500 kilometers is on average relatively low, further indicating that these kind of patterns cannot be explained by neutral evolution alone. In the next step, we further wanted to investigate how genetic differentiation is distributed across the inverted and non-inverted chromosomes. And we therefore produced phased chromosomes from locations, at 4 locations on different continents, where we compared inverted and non-inverted chromosomes from one population and compared these further to non-inverted chromosomes in large geographic distance. And as you can see by these lines, and here black always indicates the comparison of inverted and non-inverted chromosomes from the same population, you can see that the inversion really contributes most to the genetic variation in all 4 different locations that we investigated. And curiously, you can see this bath tube or suspension bridge pattern, which is consistent with the theory that recombination suppression is strongest at the breakpoints and decays towards the center of the inversion, because their double recombination may lead to a limited amount of genetic exchange between the different karyotypes. Importantly, what we also see here, if you look at this gray line, here in Europe, but also in North America, we can see that the genetic differentiation between the standard chromosomes across the different populations is fairly low. And really the genetic differentiation along these gradients is mostly prominently driven by the inversion. We further wanted to investigate what are the functional effects of this inversion. Does this inversion contribute to phenotypic variation? Because this is really something, in contrast to the very nice butterfly wings, that we don't fully understand, yes, in this system. And as a first approach, we again focused on populations in Florida, where we isolated inverted and standard chromosomes, so guys with inverted and standard chromosomes, and we compared the transcriptome of these 2 different groups in 2 different temperature regimes at 18 and 24 degrees Celsius. And what you can see above here is, again, a Manhattan plot where each dot represents 1 single gene. And you can see here that we find significant expression differences between the 2 different karyotypes clustered within the genomic region spanned by IN3RP, but also across the whole genome, which may indicate that this inversion has very strong trans effects as well. Importantly, as already indicated up here, we find that there is a very strong co-localization of candidate genes that are strongly differentiated between inverted and standard chromosomes, and the localization of genes that are strongly differentiated, as shown here in this heat map. And moreover, and this is also quite interesting, we found that the strongest expression differences are found at 18 degrees Celsius, but not at 25 degrees Celsius, which may indicate that at 18 degrees Celsius, inverted chromosomes lose buffering capacities, which may provide an explanation why the inversion is relatively rare at temperate environments. Yes, I'm sorry. This is a QQ plot. And here you can see basically the p-value distribution of the 25 degrees and the 18 degrees flies or genes. And you can see here that the distribution of low p-values is much stronger shifted to low p-values in the 18 degrees compared to the 25 degrees Celsius. Furthermore, we also investigated phenotypes, and we wanted to know, besides just expression patterns, do we also find differentiated phenotypes? And here again, we focused on our favorite temperature gradient along the North American East Coast. And we indeed identified, as you can see on the left, on the right from your side, differences with respect to lifespan, particularly at 25 degrees Celsius, but also differences in body size related phenotypes, wing size and femur length with a proxies for body size in Drosophila. And intriguingly, you can see that the inversion contributes much more to the body size variation than the standard arrangement across the different environments. And this is in good agreement with previous data collected in Australia, where Ari Hoffman's group similarly found that the inversion strongly contributed to body size variation. So in summary, I hope I convinced you that Iron-3 is putatively of African origin. It contributes a lot to genetic differentiation among, there is pronounced differentiation among karyotypes, which contributes a lot to clinal variation in flies along latitudinal gradients. While we presented a lot of data now from specifically focusing on the North American East Coast, we knew, to be honest, much less about Europe until fairly recently. And this is really a pity, because the North American East Coast was most likely colonized mostly with European flies, and it therefore remains unclear to which extent adaptive variants in North America newly evolved there or were already existing in the European populations. And to address this question, Josefa Gonzalez, Thomas Vlad, and myself founded a consortium in Europe with the focus of bringing together as a grassroots movement labs from all over Europe, but also from other continents, to jointly collect flies in their backyards and develop standards in order to better understand the demographic history, and also to bring together labs to foster knowledge exchange and really to develop a baseline for our understanding of genetic variation in natural populations. And until now, so this has been really, really successful, I have to say. Until now, we have more than 60 labs that are involved. We have sequenced more than 200 population samples. And only recently, we took this to the next level, and we joined forces with our friends in the States with a sister consortium that was founded by Alan Berkeley and Dmitry Petrov and Paul Schmidt. And this consortium, or this project that we call DEST, Drosophila Evolution Through Time and Space, now, so this is also, these are old numbers. So we have now more than 740 population samples sequenced worldwide, where we pooled in each of these samples 10 to 40 individuals and sequenced them at the genome level. And as you can see in this figure down here, one very important aspect of this data set is also to continuously sample populations at the same location in order to better understand how seasonal variation may contribute to the adaptive process as well. And there are several populations, as you can see here, which have now been collected really almost across 10 consecutive years, which is a really great resource. So I don't have time to really talk a lot about this in this context, but let me just give you a small glimpse. So we want to use this data set to learn more about the demographic history, obviously, of Drosophila melanogaster, and also to identify candidate genes for clinal and environmental adaptation. And ultimately, and this is something I cannot tackle here, we also want to understand quantitatively how these clinal candidate genes influence phenotypic variation. So as a small teaser, in the first step, we used the 8 million SNPs that we identified throughout these previously 270 population samples and used PCA to basically summarize the genetic signatures or the allele frequency signatures in this data set. And as you can see here, the first principal component very strongly separates African and non-African populations. So this is consistent with the evolutionary history of Drosophila. But on top of that, we also see this gradient here of colors along the North American East Coast. And this is also consistent with a previously detected admixture cline that is putatively the result of historic slave trade that brought African genetic variation in historic times to the Caribbean and subsequently to Southern North America. When we focus on the principal component 3 of this analysis, you see I'm a bit cherry picking here, but I want to tell a story. So when we look at the principal component 3 in this data set, we can see that they very strongly differentiate Eastern and Western European populations. And needless to say that this very nicely coincides with several hybrid zones that have been found previously. In this slide here, I use kriging to interpolate the PC scores of the different sample sizes. And we were curious what may have caused this strong divide between Eastern and Western populations. Was it climate differentiation, post-glacial recolonization patterns, Iron Curtain perhaps, yes, that may have reduced just economic trade between Eastern and Western population, or is it the result of recent demographic changes? And to address this, we used demographic history, we used coalescent simulations in combination with approximate Bayesian computation to compare different demographic scenarios. And it turned out that the split between Eastern and Western populations clearly predated the Cold War era and was approximately 1,000 years in the past. And so this is just the beginning. We are in the process to further dive into this. If we, again, as I've shown before, investigate genetic associations with different environmental variants and predictors, in this case, again, latitude, you saw this picture before, the North American East Coast. If we repeat this basically for Europe, we see a slightly different picture, namely that the region spanned by Iron-3 is not so strongly, or we don't find such a strong clustering of candidates for clinal variation. Nevertheless, most of the genetic variants shared between the different continents are located within Iron-3. 54 genes are located in here. If we include Iron-3 are shared. If we exclude Iron-3, only 6 genes remain. And these 6 genes are associated, for example, with pesticide resistance. ACE, acetylcholine esterase, is, for example, one of the prime targets, not only of Novichok, but also of different pesticides. And so they may contribute to pesticide resistance. One usual suspect that you very often find in these types of analysis is FOXO, so forkhead box O, forkhead box protein O, to name it correctly, which is a very important transcription factor involved in the insulin signaling pathway and plays a key role in really coupling dietary conditions to the development of the flies. And we were interested to learn more about whether clinal variation in SNPs located within FOXO contribute to phenotypic changes along environmental gradients. And we therefore creatively So this work was done by Ezra Durmas, who was at the time PhD student in the lab of Thomas and co-supervised by me. And we came up with the idea, or with the thought, that we could use, we could benefit from a panel of sequenced isofemale lines from Drosophila, so-called Drosophila Genomic Research Panel, where more than 200 lines were sequenced and still maintained in the lab. And so for all of these lines, we know the allelic states at basically every position in the genome. And we first sorted these lines according to the alleles into an southern alleles, and then allowed these groups of lies to recombine for multiple generations. So basically homogenize the genetic background of those. And we then tested here. So these are our target SNPs, yes, in 2 of the replicate populations. So we again did pool sequencing, and we focused on 2 different SNPs that are southern and northern populations. You can see that they are perfectly fixed in these populations for the 2 different states, and tested these inbred recombinant, or not these inbred, these recombinant populations for phenotypic differences. And indeed, we found that there are differences in ecto-adult viability consistent with planet predictions. We find increased viability in southern populations. We find increased body size in northern populations, but inconsistent with predictions, we find that northern populations were less starvation resistant. And this may indicate that there are antagonistic pleiotropic effects associated with FOXO in these natural populations. So to conclude this part, we have produced until now the largest whole genome data set of worldwide Drosophila melanogaster populations. And these data are freely available. Everybody can use it. Whenever we generate new data, we put it online. So it's a resource that we really hope that many people can use and exploit in aspects that we haven't thought of. And we find pronounced population structure, particularly in Europe. And these demographic patterns, of course, deserve further investigation. You also find a genomic signal of spatially varying selection, and we're able, at least for some idiosyncratic cases, to at least find tentative evidence for phenotypic variation associated with these different alleles. So now I've been talking a lot about abiotic factors that strongly contribute to clinal variation, but of course, organisms don't live isolated and seldom alone, and therefore they are affected and influenced by other organisms that they live with. And this interaction can lead to a full spectrum of different interactions, from mutualism to parasitism, especially then when a given environment or a changing environment, when there are different optima in the environments of the 2 different interacting organisms. But very often these dynamics and the contribution of environmental variation to these host-symbiont systems remain still poorly understood. And in my research, I'm again using Drosophila. You see, I really love the fly. But in addition, I investigate Wolbachia, which is, besides Drosophila, also a very well-studied model, and really one of the most broadly distributed endosymbionts, which really occurs in all arthropods, but also in filamental nematodes. And it has a full range of effects on their host, which can be very mutualistic, beneficial, but also to highly detrimental, leading to reduced lifespan and so on. In some organisms, they occur as facultative or endosymbionts. In some organisms, they're absolutely obligatory, and the host cannot live without it anymore. In Drosophila melanogaster, we know at least of 5 main Wolbachia types that occur in natural populations. Out of those, WMLCS, here I highlighted in red, and WML are the 2 most common variants. Particularly WML is now found in almost dominant in all worldwide populations. But as a study by Markus Riegler in 2005 indicated, this has not been like this until very recently. So these guys compared long-standing lab strains that have been collected 100 years ago for their Wolbachia types that they carry, and they identified that all of them, or most of them, were infected with WMLCS. And only very recently, when they compared it to contemporary natural populations, they found that they were all dominated by WML. And this indicates, at least indirectly, that there was only a very recent replacement of the ancestral WMLCS by the contemporarily dominant WML variant. However, I mean, this is a bit challenging because we don't know to which extent the two different types under laboratory conditions have an adaptive effect, and there may have been cross-contamination in the lab and so on that may have contributed to the patterns that have been reported here. And there's basically only one way to really dig deeper into this, and that is look at historic samples. And luckily, only recently a data set got published in 2023, so in the beginning of this year, where researchers associated with, including John Poole and Markus Densmeyer from Sweden, produced genomic scale data from historic samples that have been collected between 1800 and 1930s from different populations in Sweden, Denmark, and Germany. And so what they did basically is they used PIN museum samples and non-invasively extracted DNA out of those and sequenced these DNA with Illumina to genome scale, at genome scale level. And as you can see here, well, it looks a little bit confused, but it's still the extraction process is the most non-invasive as you can be for these museum samples. So this was very interesting in terms of learning more about the population genetics of Drosophila, but I, of course, was more interested in the bycatch. And so I identified reads of Wolbachia in most of these samples. In some cases, as you can see here, I'm showing the relative Wolbachia titer, which is the read depth of Wolbachia reads divided by the autosomal reads of the Drosophila host. So you can see in some cases the titer is relatively low, but in some cases we see that there is even more Wolbachia reads than autosomal reads relatively. And I compared this historic data set with contemporary flies using available single individual sequencing data, but also generating new data from WMLCS infected Drosophila samples. As a first step, we wanted to classify and identify with which Wolbachia type, well, this is the main question, these have been infected. And we therefore used the contemporary samples that we have to identify Wolbachia type specific marker SNPs. So we divided again the flies into groups and identified positions that were fixed for two different variants, and then checked these positions in the historic samples. And as you can see here, we were able to identify at least one of these diagnostic SNPs in at least 11 samples. And all of the SNPs, really all of these SNPs indicate that these flies were all WMLCS, which is very well and consistent with the hypothesis in Riegler et al. 20 from 2005. We further employed a phylogenetic approach, focusing on approximately 300 SNPs scattered all over the Wolbachia genome in order to identify the evolutionary history of these flies. With this method, we were able to reconstruct the four previously identified monophyletic clusters in the WML variant. And again, we were able to show here, or consistent with previous results, that all of the historic samples cluster together with the contemporary WMLCS variants. You see that some of these branches here are fairly long. we assume that this may have something to do with the level of DNA degradation in these data sets, which may have resulted in some erroneous alleles. Since Wolbachia is maternally transmitted, it is already shown previously that it is very strongly linked to the evolutionary history of mitochondria. And we therefore further, here, this should be blue and this should be red here, yes. And some of those are actually not red because these include also all samples that were not infected with Wolbachia. But nevertheless, the story remains the same. What I want to show here is that even though we were not able to identify Wolbachia in all of the historic samples, we are able to show that most of these historic samples carry the same mitochondria as WMLCS type Drosophila, which further indicates that WMLCS was very, very common at least 170 years ago, 150 years ago. There's only one sample up there from Denmark that appears to be of the more contemporary WML type related mitochondria. And if we look at this tanglegram, that further indicates the same story. We only find occasionally that there are some swaps. In this case, I don't think that this has something to do with horizontal transmission, but rather, especially here in this historic sample, with some inconsistencies potentially due to sequencing errors and degradation of the DNA. Now the question, of course, remains: why did this replacement happen, and what are potential functional reasons for this event? So one thing that is very well established is that flies and also the proliferation rates of bacteria strongly depend on temperature. And we were first curious to understand to which extent Wolbachia potentially influences the thermal behavior and the fitness under different temperature conditions. And we first investigated if Wolbachia influences thermal preference. And we therefore built this temperature measurement apparatus, which is basically an arena where you can adjust the temperature gradually from cold to hot. And then you place the flies inside this arena and test where these flies basically rest. We invested quite a lot in testing to which extent the environmental conditions and experimental design contributed to this. And you can see we built different gradient machines that really resulted in different responses. And we also found that, for example, humidity really plays a major role, so ambient humidity in the thermal behavior of the flies. Nevertheless, in a previous study, we found that Wolbachia apparently very strongly contributes to the thermal behavior in the context of different infection types. So you see here that this is the temperature, the average temperature preference of uninfected flies, this of WML, and WMLCS, the red variant, appears to really change the behavior quite dramatically. And we assume, or we hypothesize, that this may have something to do with an aversion behavior of the flies. So it's known in crickets, for example, that they can endorse a so-called anapyrexia, yes, behavioral anapyrexia, which is a negative fever, I would say, to alleviate fitness costs due to bacterial infections. We carried out these experiments with long-established lab strains, and therefore we were not sure whether these experiments were just an artifact of our lab strains and lab conditions, and whether we find similar patterns in natural populations. And furthermore, we wanted to better understand what are the causes and consequences for these behavior. And therefore, we repeated this analysis using wild-caught flies, really, that were only in the lab for a few generations. And we found that the story is, of course, much more complex. than we previously thought. We, for example, find in this case, I need to say this, we treated our lines with tetracycline. So we first cured all the infected lines from natural populations with tetracycline. And we also, as a negative control, cured our uninfected lines from natural populations. And as you can see, we basically find, for most of the cases, no real differences in thermal preference, except for one line, from one line infected with WMLCS, consistent with the results that we found previously. So it seems like the Wolbachia infections negatively induce a negative thermal preference. We further wanted to better understand what may cause, or is there indeed some kind of tentative correlative evidence that the bacteria are costly. And we therefore investigated for these flies that we tested for thermal preference. We investigated their relative bacterial titer. And in this case, we use qPCR, where we compared the titer levels or the content, the DNA content of a host gene and also of a gene associated with Wolbachia. And what you see here is that we found for this one natural strain that was infected with WMLCS, a very strong negative correlation between relative titer and temperature preference, which is consistent with our hypothesis that flies may choose colder temperatures to alleviate fitness costs. Curiously, we find similar patterns, at least for two strains that didn't show any apparent temperature preference difference. Yes, we only find these patterns when we specifically investigate relative titers and their thermal preference. Finally, we wanted to test to which extent temperature influences the interaction between the host and the bacteria. And we therefore carried out large-scale phenotypic assays where we tested natural flies infected with the two different variants under different temperature conditions for a broad range of different developmental or juvenile and adult life history related traits. And we found that there are no effects of Wolbachia on development time, on body size and variability, but that also consistent with previous results, we find that Wolbachia may positively influence the reproduction. In this case, we find the WMLCS boost reduction in the fecundity of the females, and that this change in the fitness of the females is strongly temperature dependent. Furthermore, we found that WMLCS also positively influences the maturation or the onset of the maturation of eggs. That means flies infected with WMLCS start to produce eggs earlier than uninfected flies or flies infected with WML, but only at higher temperatures. And finally, given that we found that there are strong effects and that these effects seem to be temperature related, we wanted to ask generally, what are the titer levels, or how does Wolbachia titer change in the host, in different tissues, at different ages of the flies and at different temperatures. And what we find is that these titer levels are strongly dependent on the variant and on the age of the flies. As you can see here, the older flies get, the higher the titer seems to be, which may have something to do, we've discussed this with octomom numbers, but also perhaps with reduced capacity, buffering capacity of the aging fly to regulate titer levels. And we find that curiously in the ovaries, where it really hurts, of course, a lot, if you have high titer levels, if they have negative effects, the levels seem to be relatively strongly regulated. And with this, I want to conclude, and I hope I've convinced you that Wolbachia infections can affect the preference of the host. But this is, as I mentioned, not a general pattern and strongly dependent on the conditions, but also on the host genetic background. And we found that Wolbachia does not have effect on developmental traits of the host, and there seem to be some kind of trade-offs because WMLCS has beneficial effects on early fecundity, but only at elevated temperatures. We find no tighter differences among Wolbachia at an early age and no obvious trade-offs under lab conditions. That means we don't see a boost of adult reproductive traits at the cost, for example, of reduced lifespan. One thing that still remains to be shown is why was WMLCS replaced in nature? This is something that we unfortunately until now were not able to conclusively finally answer. And with this, I want to thank you a lot for listening to my talk. I have to thank a lot of people, especially Anton, Fabian, and Sina, who really carried out all the Wolbachia stuff that I presented, and Thomas Flood and Ezra Durmas, with whom I collaborated a lot and very intensively on the inversion part of my work, and my friends and folks at DrawsEU who generated this data and with whom we collaboratively in a collaborative fashion analyze these data sets. And of course, you for paying attention. I'm happy to take questions. Thank you.
Yeah, thank you for the nice talk. Does somebody have questions?
There's a lot of you.
Yes.
I was wondering, in the first part on the inversion dynamics, I was surprised to see that the inversions seem to be polymorphic in Africa, and also they form a cline on the Western Ghats, or at least in Western India, which to me look like the climatic conditions and the range of conditions is very different from what you would find along the more temperate climes. So
that's a great question. Perhaps it has not something to do with climate in the end, but perhaps more. So we tested a lot temperature dependent effects. We didn't really find any. So it may very well be that other factors like parasitoid load or something like this may contribute to adaptive effects. And I mean, we did a lot of head scratching what's actually going on, because usually you start with different phenotypes and then you identify, oh, it's an inversion involved. In our case, it's really the opposite. And what may be in this case is that the inversion really encodes a highly complex multivariate phenotype that has very subtle individual effects. And it may be very difficult to really disentangle what's actually what is contributed there. So that's what I think.
you have trade-offs, perhaps between the different traits encoded in the inversion, and the trade-off resolution could be different according to conditions. It could be climate or other.
That could very well be. I mean, one of that we were facing in the lab, and so studying this under lab conditions may be a one-way road or not the way, is that we actually find that phenotypic values associated with inversion, at least under lab conditions, were generally we would consider as inferior compared to the standard arrangements. So the splice were smaller, they lived slightly shorter. So there are also sometimes not significant effect, but just the trends were towards less vital, yes, phenotypes. So I think it's very complicated to address or to answer this question, investigating this under the laboratory conditions that we did.
Online?
Now we can listen online to Aneta Atlet.
Thank you. Hello, everyone. Thank you for the great presentation. It was very interesting to me,
Thank you.
especially that I'm a geographer and I'm from Poland. So my question is about this significant border in between Western Europe and Eastern Europe. You saw on one of your slides concerning the difference, geographical difference. That is really surprising. And since I'm from Poland, I can say that from my experience, it looks like the flies in Poland are concentrated since late spring to really early autumn, at the period when the temperature and the humidity is favorable for the ecosystem. The flies expect. On other topic, I'm not sure if you confirm that about the ecosystem conditions. It was interesting, the thesis, the question if the historical event, the politics could influence the richness or the divide of the types of the flies. I would say something like this, that at Eastern countries, flies were concentrated mostly at the rural region, and our farming for the long time was not well developed. So probably entomologists could confirm that for a long period of time we did not even use any chemicals to eliminate or to decrease the number of flies in our environment. Do you think that could be the reason, the explanation why it is such a significantly different in between East and West? That was surprising.
Thank you very much for your question. I start with the second one. So I think that the differentiation is not necessarily the result of spatially varying selection, but rather really a demographic event, given that we find strong differentiation between Eastern and Western population across the whole genome. So it may very well be that there may have been adaptation to different, like as I showed, to pesticide resistance in some cases, but I rather believe that most of the differentiation comes from really the evolutionary history of the Eastern and the Western populations. You were saying something about the political aspect. This is something that we were very scared or it's difficult to argue in this direction, because you really don't want to start controversies here. It is rather, I would say, rather than really the boundaries follow the Iron Curtain. Well, I mentioned this, of course. We really find that these tension zones are in line with many other post-glacial organisms, such as hooded carrion crow, house mouse, red-bellied toad, yellow-bellied toad, and so on and so on. So there seems to be, I don't think that this is a post-glacial relic, but nevertheless, it may be some biogeographic patterns that may contribute to these patterns. But and of course, we don't know to which extent historic anthropogenic influences such as the Avars and the Mongols and the Huns coming to Europe from the East may have contributed to this. What is intriguing is that the genetic variation within the Eastern cluster is very low, and that I wasn't able to show this here, but whereas we find in the Western population very well separated clusters according to their geographic region, indicating isolation by distance, we find that the Eastern cluster really forms one big cluster, which may have something, or which may indicate that there is one big founder or one big source population from which there is, for example, annual recolonization patterns. We don't know. Yes.
Mm-hmm.
I hope this partially answers your question.
It's really interesting because, for instance, for the mosquitoes, I'm a bit more interested in because of they are vectors of the diseases. This kind of barrier looks like in present time does not exist. It means that the aggressive mosquitoes like a tiger, they are entering the ecological niches more and more north. And from your results, it shows that we have a kind of conservation, if I can use this kind of expression to name what you are describing. Could it be something like this? I'm not sure.
I'm also not sure. Yeah, I don't know, to be honest. Okay.
Yes.
Thank you.
I'm sorry because my question was kind of
linked to
you
that.
anticipated in some way my question. So first of all, thank you for the great talk. It was
Thank
really
you.
interesting. And I remember that there was a professor in Italy that said that we should not make hysterical biogeography, but only historical biogeography. And it was his way of saying that sometimes you are looking for a lot of hypotheses, and then we don't really have the data to test. But I was actually thinking about exactly the same thing that you were saying, so that this pattern really reminds me about glacial animals, so post-glacial animals, like in butterflies. They showed a lot of this. And actually one kind of easy, I don't know if it's easy, but we have a lot of samples, so I guess that can be easy to test this, would be to have samples from southern Italy, which we don't have, I think, in your map. And they showed a lot in this thing in butterflies. Southern Italy is a different glacial refugium compared to Spain, so Iberia and Greece, Balkans. So if you have samples from southern Italy, I think it could be an easy way of testing this.
Totally. That's a great suggestion. Yes. So if you know people in southern Italy, please spread the word. What we also need, and this goes in the same direction, is more samples from Asia, because we only speculate in that these are of Asian origin, but where actually these flies are related to which other populations is completely unknown. So we would really, I think, also from the Middle East, it would be very interesting to get more samples to get a better understanding. And perhaps, I mean, the last glacial maximum falls slightly outside the expected first colonization of Europe. But what do we know? Yes, in terms of whether flies were nevertheless able to harvest on berries in Europe, even predating agriculture. So that's something that, of course, needs further investigation.
Yeah, I have comments in this regards too, because I was involved in sampling, and most samples from East Europe is from Ukraine, actually, and those populations are not very different, and we don't have great difference in conditions like in Europe, because in Europe, in north part, climate is different, many things are different, but in Ukraine, it's rather homogeneous conditions, and we didn't see many differences in mitochondrial level before, so it can be biased because
that's a very good point. So we tested for something like branch attraction, yes, or in PCA whether you have a confounding factor by including many samples from one population. Let me tell you, when you exclude or when you subsample to only a handful of representative, the pattern stays exactly the same. And moreover, while I fully agree that Ukraine really contributed, thanks by the way, to draw you most of the samples, it's also Serbian, Polish, Russian, Austrian samples that really fall tightly within this cluster. So it's not only the Ukrainian samples, but really everything within this blue cloud. Yes, my Austrian comes through. I'm sorry. Yeah.
Yes, but if I can say anything, that suggests a kind of a rigid at the environment. Is it possible to be this process such well conservated? I mean, such a stable at such a long period, because the end of the glaciers is around 9,000 years.
Yes, but the last glacial maximum was around 14,000 years. So, and this may have pushed the flies to the different refugia, and only after that, at least I'm not an archaeologist, and but so we assumed that the colonization happened only after that. And if we consider that the agricultural development happened more in the Middle East, one could assume that you have basically a colonization coming from the east, which should not result in this east-west pattern. But I mean, in humans, we see similar patterns. If you look at the old data from Scavalli Sforza, etc. So there is more to explore. I just wanted to show, this is the map that the PCA that I was referring to from previously. So you can really see that all of the eastern populations form this very strong cluster. And even here, I'm Austrian, so I have to show this. Even Austria, yeah, so here there's these samples that are from north of the Alps, yes, from the Wachau, very nice area. If you ever come to Austria, you should go there. And these are from Carinthia, from southern, also nice area. You should also visit this. And so it's really the Alps that separate them, and it indicates that there may be that these, although being longitudinally divided, they may contribute to this separation. So you see there are many unresolved problems here, questions.
Maybe just a quick question / comment regarding the replacement of WMLCS by WML. I was wondering whether it could be a sweep of the mitochondria actually, and not of WML, and that WML would just be hitchhiking on the mitochondria. Do you want to comment on that?
Yeah, that's a very good question. I mean, we could investigate the genetic variation in the I don't really know, yes, to which extent the genetic variation within mitochondria differs between WML and WMLCS type. But that's something that would be worthwhile exploring. Thank you for this comment, yeah.
So we switch to the second part now.
Yeah.
Volbaka. So I had one question regarding whether, so you seem to hint to the fact that perhaps the flies were modulating or regulating their infection rate using by going to colder places. So if they regulate the infection rate during, I mean, through the lifetime, if I understand well, that would mean that they start off with a more or less fixed infection titer. Like, do they how their ovules are?
They don't. I mean, I agree. It's a bit of a far-fetched argument, yes. But if so, we in this was exactly also the motivation for us to really rigorously investigate titer variation throughout the lifetime of a fly in different tissues. And so what you see is that the titer variation, at least under our laboratory conditions, really seem to matter most at the later life stage. And so this one way, what could happen is that this influences also the way flies choose their breeding grounds or the place where they lay their eggs and so on. And so we don't know to which extent such a alternated behavior may contribute also to the fitness of the fly and perhaps also of the next generation. That's also something that we intended to explore further.
And could it be that rather than the fly trying to modulate the
Yeah.
motor, could it be the Wolbachia influencing the fly?
Yes, yes, yes. So that's even more difficult to investigate, I would say. So what we were initially thinking, so that was my plan when I applied for this, that funded this, was to test to which extent there is a functional interaction between Wolbachia genes, gene products, and the thermal sensing apparatus or mechanism in flies, which is quite well understood. So there are some channel proteins, TRPA1, for example, that is very well known and sensitive, located in areas where also Wolbachia is found. So, I mean, that would be one way to investigate whether increased bacterial expression may trigger behavioral changes. But we had problems with this, let's say.
Okay, thank you. And I think we don't have time for more questions. And thank you for the nice discussion.
Yeah, thank you very much.